Hello anatomy students, this is Dr. Allis. In this short video, we're going to explore a genetic condition known as sickle cell anemia. When we talk about sickle cell anemia, this disease gets its name based on the shape of a patient's red blood cells. Now, we're used to drawing salty banana cells that look like this, but in actuality, normally your red blood cells are large flat discs. This shape, or this anatomy, is really good at doing two things. The first thing it's really good at doing is transporting oxygen. The second thing it's really good at doing is squeezing into tight spaces. In individuals with sickle cell anemia, whose red blood cells aren't flat discs, we end up with these sickle-shaped cells, these curved cells. Not only are these cells worse at transporting oxygen, they're also worse at squeezing through tiny spaces. This is why patients with sickle cell anemia can have things called crises, where literally their blood gets cut off to certain parts of their body because all the sickled red blood cells build up in one area. Let's talk about the genetics that leads to sickle cell anemia. Patients with sickle cell anemia have a mutation in one nucleotide in their genome. Notice that an individual without sickle cell anemia has the codon CTC in their DNA. Individuals who have sickle cell anemia have the codon CAC. Remember when we talked about the genetic code table being degenerate? It meant that we have some wiggle room. If I make an error in the process of transcription or translation, I might end up with the correct amino acid. But that only applied to the last nucleotide in a codon. When we talk about sickle cell anemia, the nucleotide that has been mutated is in the middle of the codon. So when a cell does the process of transcription, going from DNA into RNA, instead of making the codon GAG, which is translated into the amino acid glutamine, an individual does the process of transcription and ends up with the codon GUG. When GUG is translated, this calls for the amino acid valine. Not only are glutamate and valine completely different personality types, they're about as opposite as it comes. Glutamate is a negatively charged amino acid. Remember that things that are negatively charged are attracted to the positive dipole in water. When we talk about valine, valine is a nonpolar amino acid. Nonpolar amino acids don't play nice with water. They're what we call hydrophobic. So, in an individual with sickle cell anemia, we've added in a new amino acid, valine, an amino acid that doesn't play nice with water. We've added it in a place of an amino acid, glutamate, that does play nice with water. This gives us big issues as we start talking about the tertiary and quaternary structures of the hemoglobin protein. Let's start by taking a look at the levels of protein structure in an individual who has normal hemoglobin, meaning they do not have sickle cell anemia. Notice our primary structure is intact. There's no mutation in our nucleotides, so there's no mutation in our sequence of amino acids. Primary structure is the order of the amino acids. When that order of the amino acids is correct, my alpha helices and beta sheets, or my secondary structures, as well as my 3D shape, my tertiary structures, are also intact. We end up with a part of my protein that looks kind of like a heart. This heart-shaped part of my protein comes together with a few other proteins to build the quaternary structure of hemoglobin. This quaternary structure of hemoglobin is a free-floating 4-piece protein. Let's compare this normal hemoglobin that's free-floating to the hemoglobin we see in a patient with sickle cell anemia that builds long protein strands. The problem in sickle cell anemia goes all the way back to the primary structure of the protein. When we first started building this protein, instead of putting in a glutamate, we put in a valine. And remember that valine was non-polar. Non-polar means the same thing as hydrophobic. When I put in this amino acid, this valine, I generate this place on my protein that is hydrophobic. And because it's hydrophobic, it doesn't want to be around water. So I put this protein with its other proteins to build a quaternary structure. It's not right compared to my normal hemoglobin, but I've built one. But the problem is, in this quaternary structure, I have two places that don't play nice with water. And yet the inside of a cell and outside a cell is full of water. This leads my proteins to find each other and connect. Remember from lesson number 5, we talked about hydrophobic attractions. We all hate water, so we all stick together. This is where these long proteins come from in sickle cell anemia. I hate water up here, you hate water up here. If we squeeze our water-hating parts together, nobody's by the water and everybody's happy. Everybody, that is, except the red blood cell, that's now full of these hemoglobin proteins that exist in lines instead of free-floating inside the cell. So big picture, when we look at cells that are normal red blood cells and cells that are sickled red blood cells, it all comes down to what's happening with the hemoglobin proteins. If they're free-floating as they normally should, they'll be really good at attaching to oxygen and really good at allowing this red blood cell to change its shape. If those hemoglobin proteins are all clumped together, like we see here in sickle cell anemia, that makes the membrane of this red blood cell stretch out in incredibly unhelpful ways. It also decreases the efficiency of this hemoglobin at transporting oxygen. So sickle cell anemia is a genetic condition that starts with a 1 nucleotide mutation. If I change the sequence of the DNA, I change the primary structure of my protein, which changes the secondary and tertiary structures, and ultimately leads to a difference on the quaternary structure level. And that's why these red blood cells have such a funky and unhelpful shape.